Hello and welcome back 365 class. Today we should be covering those products of photosynthesis. So the allocation, translocation, and partitioning of the photoassimilates, which is covered in chapter 9. Now, I'm sure you are aware that there is a 24-hour curfew, so more details on Tribune 242, and I'm sure you were all listening. So, please try to stay safe, especially try to avoid big crowds and try to avoid being around people who were, traveling or who were around big crowds because the virus can stay inside the system for almost 2 weeks. You would not have any symptoms. However, you can still spread the virus and other people can spread the virus to you. So I know these are very difficult times, but I have to still keep trying lecture and do everything on Moodle. So this is one of the chapters that is going to be on the essay tests, which will be on April 4th. Okay, so let's slowly start talk about that. Okay, so we said that in most plants, right, during the day, this is where we have in the chloroplast the electron transport chain on the granium, and then we have the Calvin cycle where CO2 is being absorbed, and then in the end we will see that there is that there are those trial sugars which are being accumulated. Okay. Now, what we have also mentioned that inside the chloroplast, there has to be a large pool of those triophosphates. So only some of those will be exported out. So pretty much with those trial sugars that are being exported, okay, that do not remain in the Calvin cycle to replenish the RUBP, they can have two pathways. So they will be either converted into starch for storage, or they will be exported out of the chloroplast and then assembled into hexose sugars, mostly in the form of sucrose. So sucrose is a form of sugar that can be exported out from the cells that have chloroplast, and they can photosynthesize onto other types of cells. So that is what is happening during the day. During the night, those starch that has been accumulated is going to be divided into either glucose or maltose, and we need those two types because we only have glucose and maltose exporters. So these are transmembrane proteins on the chloroplast, and once we export those, then they are assembled and converted into sucrose, which can be exported out. So during the night, even if there is no light, of course, and the stomata are closed, there will be some sugar transferring across the plant based on the starch that is being stored. Okay, so I know this picture that I have right here is it is kind of bad. It's from an old book, but it is showing us very well what is happening. So that sucrose is then going to be transported, and I quote, over long distances, used for, let's say, growth at the stem or root or new leaves, new flowers, or it is going to be stored in grains and tubers, or there should be another arrow right here. So there is another type of sinks. Usually these are fruits and different types of vegetables. So yeah, pretty much this is what I just said in text. The production of starch and sugars, that is called carbon allocation, right? So let me just repeat one more time. Starch is a form of storage of carbon, while sucrose is a form that carbon is being transported over long distances in plants. So let's talk about starch biosynthesis. So as you can see from the previous picture, starch is being synthesized in the stroma, in the chloroplast, mostly in leaves, in that palisade mesophyll. There are two types of starch, amylose and amylopectin. One is branchy, and the other one is the amylose is more linear, and also the difference is those connections. So the glucose molecules are connected with the alpha 1,4 linkages, and the amylopectin is connected with the alpha 1,6. Don't get confused. This means that at the branching point, with the amylopectin, we have the first and the sixth carbon that are being connected, and here we have the first and the fourth carbon from the glucose, which are being connected. So this is amylopectin and amylose. And of course, glycogen, which is even more branching, is the storage material of animals. There are two key enzymes with the starch biosynthesis called ADP glucose pyrophosphopyrolase and starch synthase. So here you can see the accumulation of starch granules inside the specialized cell called the amyloplast. So what are the steps in the starch synthetase? So let's start talking about that. Now, all the G3Ps are being converted into dehydroxyacetone 3-phosphate, which is a form of triosugars which can pass through any type of membrane. So all the 3PGs which are being synthesized by the end, in the middle of the Calvin cycle, have to be converted to dihydroxyacetone. Then dihydroxyacetone is connected with glyceraldehyde triphosphate to form fructose 1,6-biphosphate. So for those of you who are not a chemistry bugs, you know fructose, it has 5 carbons, and then 1,6, it is showing you that the phosphate group are the phosphate groups, so the 2 biphosphate groups are on the first and the sixth carbon. So the first and the sixth carbons are those that carry those phosphate group. Then fructose 1,6-biphosphate is converted into fructose 6-phosphate. So we detached one. Okay, so we detach one phosphate group, and then we converted that molecule. So we converted fructose 6-phosphate into glucose 6-phosphate. Then in the step number 4, we converted glucose 6-phosphate into glucose 1-phosphate. So the plant, using this enzyme, phosphoglucomutase, just shuffled this phosphate group from the position 6 to position 1. So from the 6th carbon onto the 1st carbon. Now, this end step is extremely important. What is going to happen is now this molecule of glucose 1-phosphate. So this is a molecule of glucose that has a phosphate group on the first carbon. We are going to add ATP. Actually, we're going to add a ATP molecule onto that. Okay. This labeling of molecule of glucose is extremely important. You don't really have like a free-floating molecules of glucose in the cytoplasm. Every glucose molecule knows where it is going to go. So if this glucose molecule was, fated to be a part of starch, then you're gonna add ATP onto that, and that's like a flag. Okay, put this glucose molecule in starch. Like I said, starch, it's just like glucose molecules connected with the oxygen bond. So only glucose molecules that have ADP onto them will be added to the growing starch chain. Okay, please remember this because you will see what is going to happen next in the sucrose synthesis.